Hey, thanks for coming. This talk today is going to be recorded for folks down at Texas A&M, students that belong to Professor Michael Speed's class, and so I thank him for asking me to do this, and corresponds to a talk I gave at Texas A&M a few years ago, and it's quite appropriate for my current class here at Kansas State. So I have 6 students in the room that have been bribed to come to class today on a Friday afternoon, starting about 4:45. So you can see we have dedicated students here at Kansas State University. I have some introductory remarks to start with, and then I have some examples that I wanted to show you some of the different kinds of messy experiments. I call them messy because I wasn't quite sure how to analyze them when they first showed up. And so I wanted to show you those and help you think about or see the way that I try to look at experimentations. So to begin with, though, is a more generic thing. Suppose that one asks, why should experimenters or researchers be interested in statistics? And the basic answer is reliable facts are necessary to build a correct science or theory, and reliable facts are often not easy to come by. And statistics and the design of experiments are concerned with procedures for obtaining reliable facts. There are basically 3 phases of experimentation: formulating the experiment, designing the experiment, and then analyzing the data that results from the experiment. And I often like to ask folks, which of these phases do you think a statistician is most useful? For many researchers, the answer would be different than it probably would be for most consulting statisticians. Many researchers think the statistician is most useful when it comes to analyzing the data, but most statisticians think they're most useful when it comes to designing the experiment. If you design the experiment properly, then the analysis will pretty well take care of itself. And it's also amazing how much input a statistician can have when it comes to formulating the experiment as well. In formulating the experiment, the basic questions that one looks at is, what are the problems to be solved? What is the experiment intended to show? What kinds of hypotheses will be tested? What variables are going to be measured? What factors are going to be varied? What is the treatment structure? Is the objective to show things are different or to show things the same? This is a very important question, because if you're going to show things different, then you can do that with a much smaller sized experiment than you can if you're going to try to show things are the same. And so that's an important consideration in formulating the experiment. In designing the experiment, we're interested in such things as, what are the experimental units? What are the measurement units, perhaps? Are missing values likely? Why or why not? How many experimental units should be used? That's a sample size question. How many replications? Are there ways that the experiment, or how should the data be collected? Are there ways that the experimental units can be blocked? Should the experiment be done according to an experimental plan? How should the randomization of experimental units to treatments be done? What kinds of assumptions might be reasonable? How accurate is the experiment likely to be? What models are likely to be most meaningful for the experiment? What are problems that might occur? Can the data that is collected be analyzed? Will the data answer the important questions? And are there independent replications? Convenience often destroys many experiments, or limits the amount of information that can be gained from an experiment. And so one has to be very careful about doing things in the most convenient way possible, because you may end up with information that's not very useful at all. I remember when I was a graduate student, I was helping a researcher who was trying to study the relationships between people that were in nursing homes and their quality of life type characteristics, and the amount of food waste and type of food waste there might be on plates. And so this particular researcher got individual data on each individual nursing home resident, but then when they did the food waste study, she just pooled all their information together, cleaned off the plates as they came in, and then analyzed that. So she basically didn't have any individual data on one of the characteristics that was most important to her to look at. When it comes to analyzing the experiment, some of the important questions are: what statistical tests are likely to be appropriate? Can the design be analyzed by a statistical package? If so, how should the data be organized for computer entry? Will one be able to communicate these results to others? Are the assumptions that you're going to make likely to be satisfied? Can they be tested? And can the data still be analyzed if the assumptions are not satisfied? In selecting a good design, want to try to keep the design simple, use blocking effectively. Smaller block designs are more efficient than larger block designs. Incomplete block designs are usually more efficient than complete block designs. And make sure the design can be analyzed, and can it still be analyzed if there's missing data. And then some examples of what we might call simple designs. Maybe they're simple to some of us, not so simple to others just yet. But completely randomized designs, randomized complete block designs, Latin squares, crossovers, Greco-Latin squares, incomplete block designs, split plot designs, strip plot designs, and many confounded designs that might be variations of those. Some general comments. Think about experiments in terms of treatment structure and design structure. Almost any treatment structure can be studied with any design structure. Be sure to know and understand the difference between independent replications and dependent replications. The analysis of the design structure provides estimates of different error variances that may occur in an experiment. And the analysis of the treatment structure, the hypothesis to be tested, is similar for all design structures. And it's important to remember that it's usually no harder to conduct an experiment with a well-designed plan than it is to conduct an experiment that is not well-designed. The folks here at K-State will have seen this example before, but I think it's an important one that tries to address the issue of whether you have independent observations or dependent observations. And so suppose we want to compare the heights of black males to white males, and in some population, we select one male from each population of blacks and population of whites in whatever, well, maybe we're doing this at K-State, and we have 10 people measure each person's height. We denote the data by Xij as I goes from 1 to 2, and J goes from 1 to 10. We compute a t-statistic, usual t-statistic, and if the t-statistic is large, we decide that there's a difference, significant difference between black males and white males. But the important thing here is that while we have, in some sense, we have 10, the people that are making the measurements are probably not correlated to one another. One person tends to measure low. That doesn't mean the other person is going to measure low. They might actually measure high. And so there's independent observations with respect to the measurers, but we don't have independent samples from the population to what we want to make an inference. So the important thing is that we get independent data, independent observations from that population that we want to make an inference to. And most people that would see this example would easily identify this as being a very dumb thing to do, and nobody in their right mind would ever do it. And I agree with that, but I've seen people in their right minds do things very similar. And this is an example. Suppose we want to compare the effects of storage temperature on the quality of CDs or DVDs. We randomly assign a high temperature and a low temperature to each of 2 storage chambers. 10 CDs are placed in each chamber. Then 3 months later, the CDs are removed and measured for quality. And so we denote the data by the Xij's again. We do a t-statistic, reject the hypothesis of equality if the absolute value of t is large. And so here we have the same problem. The variability that we're measuring is not variability between storage chambers of different temperatures, but we're measuring variability within the storage chamber. And there's other things that could go on with different storage chambers that are not being incorporated into this particular t-statistic. And so this would be a very bad way to do such an experiment. The next thing I want to do is, and as I said, all that previous stuff are things that you should think about before you start the experiment. Bad time to think about some of those things is when the experiment's already been done. Unfortunately, we as statisticians don't often get to think about things until the experiment's been done, and then somebody brings it to us to try to analyze. And some of the things I'm going to talk about next are some of those kinds of experiments that I've come across. The first example, though, is suppose an experiment is going to be conducted to study the effects of baking temperatures and surfactants on bread quality. 4 different baking temperatures are to be used in conjunction with 5 different surfactants. We might denote the temperatures by 300 degrees, 350, 400, 450, and denote the surfactants by C for control, and then S1, S2, S3, and S4 for some different types of active surfactants. 2 loaves will be made for each of the 20 combinations of the temperature by surfactor combination. So the treatment structure here is a 2-way. We got 4 levels of temperature crossed with 5 recipes. And the question might be then, how should the experiment be conducted? And there's times where I've spent a whole hour talking about this with students, and I don't have time to do that today. But it's an important, it's an interesting thing to consider, because no matter what you know, everybody knows something about baking. Even if we've never baked. I've never baked a cake myself or baked a loaf of bread. Well, I have baked a loaf of bread in the bread machine, but most of us have never baked anything before, but we can at least think about baking. We watched our mothers bake and so on, so we have a little bit of idea how baking goes on. And so some possible designs that one might use here is a completely randomized design. And if we're going to make 40 loaves in a completely randomized design, then we have to do 1 loaf at a time. And we have to choose which one we're going to do randomly from the combinations of 20 recipes to use. And so it's a very inconvenient way to do the experiment, because it takes a long time. You've got to have 40 different bakes, you've got to mix up 40 different batches, and it's going to take you a long time to do that experiment. Randomized complete block design probably isn't much better. We might be able to do 20 loaves this week and 20 loaves next week, so we use week as a blocking factor, but it still takes 40 bakes and 20 individual loaves, 20 individual ovens, and so on. And so a split plot design would maybe one could have ovens big enough that it would be able to put 5 loaves in at the same time. Then we could do the baking thing all at once, and that would save a lot of time. And so that would create a split plot experiment with temperatures assigned to the oven, so they become the whole plots, and the loaves of bread within each oven is becoming the subplot units. And then we could also do some mixing. We could mix doughs, mix enough dough so that we get 4 loaves out of 1 mix. And then that might be another way to save some time. Rather than mixing the loaves individually, we could mix 4 loaves at a time and maybe create a split plot design with the batch being the whole plot. Or maybe we do both, the 3 and 4, and create a strip plot design. And many other designs would also be possible. One of the first experiments that I saw when I came to K-State 30-some years ago was a researcher over in entomology wanted to study the effects of different levels of green bug infestation on 3 varieties of sorghum. And what he had done is he created some, using large coffee cans, the 3-pound coffee cans, and you can think of these as being the lids as you look down on top of the coffee can. And he drilled 10 holes around the top of the coffee can, and then he was going to put a different test tube in each hole. And in those test tubes he was going to grow one of these green plant, or one of these sorghum varieties. And after the sorghum variety got growing pretty good, then he was going to infest them with green bugs. So he was going to put 0 green bugs in one of the test tubes, 10 green bugs in another, 20 in another, 30 in another, 40 in another, and so on. So he had 5 different levels of green bug infestation that he was going to use. The three varieties were a control variety, and then there was a variety, he called it E+, that had one gene in the variety that was supposed to be resistant to green bugs. And then he had some another variety that was double, or had two genes for green bug resistance. So the three varieties are control, E+, and E++. And when he first came to me, this is the way he had the experiment designed. He was going to take one of his coffee cans, assign the control plants to it, another coffee can would get the E+ plants, and a third coffee can would get the E++. And then he was going to use two test tubes in each coffee can, give two of them the low level of infestation, two of them the medium or 10 level, two of them the 20 level, and so on. And then do that similarly for all three cans. And then these three cans were going to, or coffee cans, are going to be placed in a growth chamber and observed at some time later, at some later point. And then this was going to be replicated 4 more times, giving 4 complete replicates, so it'll be replicated in 3 more growth chambers. And so if you look at the ANOVA table there, what you have is a split plot design. The coffee cans are the whole plot units, and the levels of, and the varieties are assigned to those. And the levels of infestation are subplot units. The test tubes themselves are the subplot units, and different levels of green bugs are assigned to the test tubes. And so if you look at the ANOVA table, in this case, you have what we might call as a between container, between coffee can analysis, and a within coffee can analysis, or test tube analysis and coffee can analysis might be another way to denote those two things. And so in this particular case, the thing that's most important to test in this case is there a difference in varieties. It's thought if you put 40 green bugs in one of those test tubes, you're going to get a lot different response than if you have no green bugs in there. So the level of infestation was probably going to be pretty dramatic, but the differences in varieties would be relatively small. And so this is a very bad design because it doesn't give you very much information about varieties. You only get 6 degrees of freedom to test whether there's a difference in the variety means or not. So I suggested that he use an alternative design, that he use a balanced incomplete block design, take one of his coffee cans and put both the control and E+ in it, take another one, put the control and E++ in it, and a third one, put the E and the E++ in it. And then there's 5 test tubes that have control over here and 5 test tubes that have the E+, and so then you can put in your 5 levels of green bug infestation within each of those coffee cans. And so this sort of suggests doing that where the different colors here correspond to different varieties. And it shows in the first one here how the green bugs might be, the levels of green bug infestation might be randomized to the test tubes in that thing. And so if you look at the analysis of variance for that, then the comparison between varieties now becomes also a subplot comparison because I have both varieties in the same coffee can. So now I got 82 degrees of freedom with which to compare whether there's a difference in varieties or not. And that should give you a much more powerful test than one based on 6 degrees of freedom. And we get the same, we really don't lose any information on the levels, and we don't lose any information on the variety by level interaction. I guess our error degrees of freedom dropped from 96 to 82, but 82 is still pretty darn big and should give good results. I have to say that I was really proud of this design. I was running around all over the department showing all the faculty members and all the students this great design I developed and how we were going to use it, and it went along fine till the guy brought the data back. And then turns out when he brought the data back that when he put 40 green bugs in a test tube, they ate all the plants. So there was missing data. There wasn't any data to observe. And even with the 30, they ate most of it, so there wasn't much there. And he noticed that in Rep 1. So in Rep 2, he didn't use 40 anymore. He didn't use 30. In Rep 3, he didn't even use 20. So I paid for it when it came time to do the analysis. And back 30 years ago, we didn't have SAS. And so the data analysis was pretty bad. But now it wouldn't be so bad to handle that experiment. This is an example. I don't remember where this example came from, but the individual had 4 chemical treatments, 2 irrigation levels, and 2 cultivars, 2 different kinds of varieties. We're going to be studied using 4 growth chambers, and he had 2 pots of plants of the same variety, same irrigation treatment, same cultivar placed in each one of those little squares. And so for convenience, so he didn't lose track of what was what, he arranged the growth chamber so it was very convenient to look at. He had chemical 1, chemical 2, chemical 3, chemical 4 going across the top. Had the back ones were no irrigation, the front ones were irrigation, and then cultivar 1, cultivar 2, cultivar 1, cultivar 2, and then the 2 pots within each plant. So we had 2468 times 4, 32 observations per growth chamber. 4 growth chambers, he had 96 data points. You thought, wow, I got a good experiment, but there's no randomization anywhere. So whoever did this would have to come to terms where they waste a lot of time because there's no information in that particular experiment without any randomizations anywhere. Another experiment I came across one time, I went to Kansas City with some folks, and we went to a pharmaceutical company in the Kansas City area. And they had a laboratory where they were doing an experiment with dogs. And they had a room full of cages, and they had dogs in these cages. And they explained the experiment to us, and each cage had 10 dogs in it. These were big cages, so the dogs could run around in there a little bit. And they wanted to study the effects of different diets on these dogs. I'm just showing 3 cages here, but there are 5 diets, and so there were actually 5 cages that were involved. But unfortunately, they had assigned diet C to all the dogs in cage 1, diet E to all dogs in cage 2, and diet B to all dogs in cage 3. And so there wasn't. So the cage becomes the experimental unit. The fact you've got 10 dogs in there doesn't help you too much. You get the average of 10 dogs, but you get 1 measurement per cage that you can analyze. So it didn't look like a very good experiment at that point in view. But even worse, found out that each rack of dogs was measured once a week for several weeks, and Diet C always got measured on Monday, Diet E got measured on Tuesday, Diet B got measured on Wednesday, and so on. And so the diet effect was completely confounded with the day of the week effect. You wouldn't be able to separate the two. And furthermore, when you walked into this room, these dogs were really happy to see you. So they would run around, they'd jump, jump up and down and everything. They were so happy you were there. Somebody was paying attention to them. And I know that if on Saturday and Sunday no one was in the laboratory, then the dogs would be very, very quiet. There wouldn't be much to excite them. And so the dogs you measure on Monday, I know, are going to be a lot different than the dogs you measure on Friday, even if they had the same diet. So it was a very bad experimental plan. I told them so, and they weren't interested in my information. This example is a data set that was brought to me by the head of the department here at K-State, probably about 26 or 27 years ago, but it's a lot of fun to talk about. The experiment was conducted to study 6 herbicide treatments, 4 wheat varieties, and 3 nitrogen levels. So the 6 herbicides are shown there. The wheat cultivars are Newton, TAM 105, Eagle, and Larned. And for those of you down at Texas A&M, you probably recognize TAM 105 as being a variety that was developed at Texas A&M. I'm sure the Newton and Larned varieties were developed in Kansas, since we have towns with those names. I'm not sure about Eagle. And then it also had 3 nitrogen weights that they were going to study. So we got 6 times 4 times 3 is 72 treatment combinations to consider in this 3-way treatment structure. And so a field plot for the way the experiment was going to look was like this. This is a field that has 72 plots in it. If you count the little rectangles there, there's 72 of those. And there were 2 more reps that look just like that. And if you're going to randomly assign those 72 treatment combinations at completely at random to each of the plots to develop a randomized complete block design, then it would be very inconvenient to actually do the experiment, because you'd always have to be changing your nitrogen level, you'd always have to be changing your variety that you're planting, you'd always have to be changing the herbicide that you're going to use. And so they didn't do the experiment in a randomized complete block fashion. They did it in a manner that was much more convenient, in that all the plots that occurred up here and then over here and so on got the same variety. So Larned went to the first, was selected at random from the 4 varieties that got assigned to the first group. Newton got assigned to the second group. TAM 105 got assigned to the third group, and so on. So one of these things becomes the experimental unit to variety, for variety. That's what the varieties were randomized to. And so, well, getting a little bit ahead of myself. So that's the way the varieties were assigned. The nitrogen levels were assigned going in perpendicular to that, so that all these plots got 80 pounds of nitrogen, all these got 0, and all these got 40. And then when they got to the herbicides, and they put all 6 herbicides, they went in strips down this way. So this first long narrow strip got herbicide 1 or 5, the next long narrow strip got herbicide 4, and so on. Then a new randomization would be done within each of these remaining columns in each rep. So how do we analyze that data? That's the question my department head asked me at that time. And so here's the setup here, and with the varieties assigned, the 4 varieties assigned to those columns. And so if you just had that experiment, then that's a pretty simple experiment. You got a randomized complete block design with 4 treatments and 3 blocks. And so if we were doing that analysis, then the experimental unit for variety or cultivar is one of those long rectangles, large rectangles, and so we'd have 3 blocks or 3 reps, so we have 2 degrees of freedom, and 4 cultivars, so there's 3 degrees of freedom for that. And then the error always comes from the design structure by treatment structure interaction. So the design structure is a randomized complete block, the treatment structure is a cultivar, and the interaction of those two forms the error for that particular experimental unit, or for cultivars in this case. And a similar thing would go along if you were looking at the nitrogen treatments. Again, if you just had nitrogen only, then it's a simple randomized complete block design with 3 treatments and 3 blocks. And so the analysis for one of those experimental units is just a simple randomized block analysis with 2 degrees of freedom for blocks, 2 for nitrogen levels, and the error for that would be their interaction with 4 degrees of freedom. So if you put those 2 tables together, then you have this much of the experiment worked out. You got reps with 2 degrees of freedom. It's the same for both cultivar and nitrogen. Then the cultivar by nitrogen part shows up here, and the nitrogen part up here, down here. Now consider a cultivar by nitrogen interaction contrast. So if we think about nitrogen and cultivar both at the same time, and we look at an interaction contrast, then this shows you an interaction contrast between nitrogen levels 0 and 40 and cultivars Larned and Newton. And so you can see that an interaction contrast involves experimental units of this size. And so we need that going into our analysis. And so we'll have a cultivar by nitrogen interaction with 6 degrees of freedom. And the error for that comes from the interaction between the treatment structure and the design structure. So we take the cultivar by nitrogen interaction with 6 degrees of freedom and take the interaction between it and replicates with 2. And so 6 times 2 gives us 12 degrees of freedom for that particular error. And this design, now that you look at it in both ways, is called a strip plot design. We've stripped nitrogen treatments and we've stripped the variety treatments. Okay, now look at the herbicides. And if we look at a herbicide contrast, if we want to compare herbicide 1 to herbicide 6, then we would have, we'd take where herbicide 1 is plus and the minus here, and then if herbicide 1 was here and herbicide 6 was here, we'd have a plus minus there, and herbicide 1, herbicide 6 here. And then we'd add that, do that same thing across all 3 reps. But the experimental unit for the herbicide main effect contrast is one of these long rectangles. And if you look at the herbicide by cultivar interaction, this represents up here represents a interaction between herbicide 5 and 4 and the varieties Larned and Newton. And so, and we'd have that again in the other 3 reps. But you can see that interaction contrast also involves those long, narrow rectangles. And so the experimental unit for herbicide and herbicide by cultivar are those long rectangles. And so we can add that piece into our NOVA table. We have herbicides with 5 degrees of freedom, herbicide by cultivar interaction with 15. And then the error comes from pooling together the herbicide by rep and the herbicide by cultivar by rep. And so herbicide by cultivar by rep has 5 times 15 times 2, so there's 30 degrees of freedom there. And rep by herbicide has 10, and so 30 plus 10 comes up to the 40 that we see there in that error. And then finally we consider a nitrogen by herbicide interaction contrast and a 3-way nitrogen by herbicide by cultivar interaction contrast. So if we look at this over here represents a herbicide by nitrogen level contrast. So herbicides 5 and 4 with nitrogen levels 0 and 80. And then we could have that in each of the other varieties and each of the other reps. That same kind of a contrast. So the experimental unit corresponding to that kind of contrast is one of these little fellows. That particular size of experimental unit there. And if you look at a 3-way interaction contrast, here's an interaction contrast between 2 herbicides and nitrogen and TAM1, and then you take the difference between that and the same contrast over here for Eagle, then that would form a 3-way interaction contrast. And so the 3-way interaction contrast, it also involves these little fellows. And so the experimental unit size for both the 2-way interaction contrast and the 3-way interaction contrast involves that size of experimental unit. So we're going to have a herbicide by nitrogen with 10 degrees of freedom, a herbicide by nitrogen by cultivar with 30 degrees of freedom, and then the error comes from pooling those 2 with reps. So herbicide by nitrogen by rep gives us 20, herbicide by nitrogen by cultivar by rep gives us 60, and 20 plus 60 is the 80 that we have down here. So sort of summarizing, we have 5 different experimental unit sizes going on here, 5 different sizes of experimental units to consider, and each size has its own error part in the ANOVA table as shown in that particular case there. Now, right now my record for the experiment that has the most error terms in it, I'm up to 9. I haven't seen anything yet that has more than 9, but seen that at least once. Here's another example. This experiment was conducted to determine the effects of protein and energy levels on the growth rate of lambs. And so there were 2 levels of protein, high and low, and there are 4 levels of energy. Labeled here as E1, E2, E3, E4. And the E1 and 2 were not the same, but they were both lower than the E3 and E4, and E3 and E4 weren't the same, but they were both high. And each of the 8 diet combinations were randomly assigned to 2 lambs selected at random from a group of 16 lambs. Data was collected for a week, and then each lamb that received E1 during the first week received E2 during a second week, and those getting E2 the first week received E1 the second week. And similarly, each lamb that received E3 during the first week received E4 during the second week, and those getting E4 the first week received E3 the second week. So the design looks something like this then. The 8 lambs on the high protein, 8 on the low. 2 lambs out of each group got E1 the first week, 2 lambs got E2, 2 lambs got E3, and 2 lambs got E4, and so on. And then when they switched over, crossed over the second time, they didn't do a complete crossing because there was no E1 followed by E3 or no E1 followed by E4. E1 was always followed by E2, E2 was always followed by E1, and E3 was always followed by E4, E4 was always followed by the E3. So then the question is, how do you analyze such an experiment? And so suppose we going to consider this just by looking at what linear combinations of the observations corresponds to each of the effects. So suppose that lamb here is identified by I, protein by J, energy level by K, and time by L. So if you tell me I, J, K, and L, I can tell you where I'm at in this table. And so the data in this column would be, let's say, be noted by I, J, K1 at time 1, and over here Y, I, J, K at time 2. So those are sort of the observations that occur in each of those columns. So if I'm going to look at the protein main effect, I'm going to add over all observations where proteins at the high level and subtract all observations where proteins at the low level. And so that represents that particular contrast that's shown here. So I got pluses on all 8 data points where protein is high and minuses on all data points where protein is low. And the important thing to note about that in this particular case is that's what we call a between lamb comparison, because the lamb effects don't cancel out. Lamb 1 occurs both in both times, and so well, as I said, we would call that a between lamb comparison or between lamb contrast. If you consider energy levels, then energy levels we have say if we were comparing energy 1 to energy 2, then we would have a plus minus here, minus plus here, plus minus here, minus plus here, and so on, or as shown on this next transparency or slide, we would have this particular contrast. So that contrast is we have a plus and a minus on each lamb, so the lamb effect cancels out, and so we call that a within lamb contrast. And if we were comparing E3 to E4, then it would be similar. That would also be a within lamb contrast. There's 4 energy levels, so there's 3 degrees of freedom for comparing energy. These 2 contrasts, energy 1 versus energy 2 and energy 3 versus energy 4 are orthogonal to one another. And so the other contrast that's orthogonal to that would compare the 2 low levels of energy to the 2 high levels of energy. And so if you do that, though, that particular contrast is between lamb contrast, as you can see by not having pluses and minuses in the two time periods for each particular lamb. Well, if you continue this, if you look at a time contrast, time 1 minus time 2, so we got pluses all everywhere at time 1, minuses everywhere at time 2. That's a within lamb contrast. If we look at protein, say an interaction, protein crossed with the low energy contrast, where we compare E1 and E2 and protein high and protein low, so that two-way interaction contrast would be estimated by the linear combination illustrated there, where I didn't put the zeros in, but just the cells that have either a plus or a minus on them. And that's a within lamb contrast. Protein by combined energy. And this one here would be the protein by high energy, would be similar to that. Protein by combined energy is shown there. And you can see that that's a between lamb contrast. If you look at protein by time, the contrast is shown there. That's a within lamb contrast. If you look at low energy by time, then that's a between lamb contrast, as would be high energy by time. If you took combined energy by time, then that's a within lamb contrast. And the three-way interaction, protein by low energy by time, ends up being a between lamb contrast, as does protein by high energy by time, and the protein by combined energy by time ends up being a within lamb contrast. So if you key out what the ANOVA table is going to look like, we're going to have the ANOVA tables partitioned into two parts, between lamb analysis part and within lamb analysis part. In the between lamb analysis part, we have those effects that are that have pluses on both times, and the within lamb analysis part is the part that has one plus and one minus for each of the two time points, so that the lamb effect cancels out. Now, the problem with this one is writing a computer program that will give you the test. So it's kind of an interesting thing, because you got these three levels of energy, but it's partially confounded with lambs and partially not confounded with lambs. So 1 degree of freedom is confounded with lambs, the other 2 degrees of freedom are not. And so I'm not quite sure how you write a computer program to do that. So you might have to do this analysis by hand, even with today's software. The last example that I have is experiment was being conducted to compare 3 treatments on pigs. The treatments were, I call them A, B, and C, but A was uninfested pigs, B was infested and treated, and C was infested and untreated. The experiment was conducted on 24 male pigs and 24 female pigs. The 24 pigs of each gender were assigned to blocks according to weight. So there were 3 pigs in each block. So there's 8 blocks of male pigs and 8 blocks of female pigs. 2 pigs of each gender which have been assigned the same treatment are placed in the same pen. So there are 12 pens, each containing 4 pigs, 2 males and 2 females. And because of the nature of the treatments, the pigs that are assigned to the same pen must all receive the same treatment. So the uninfested pen pigs in that pen have to be uninfested. You can't infest some and uninfest some, because it'll transmit to the ones that are uninfested. So they have to be penned separately. And then measurements are taken on each pig weekly for 5 weeks. And the question is, how should that data be analyzed? I tell people that this is an experiment that I use bootstrapping on. I had the researcher in my office. I took my bootstrap off the wall and I threatened him with it. I said, if you ever run an experiment like this again, I'm going to whop you with my bootstrap. So I don't know whether you think that's funny or not. Okay, so in this particular case, the the only fixed effects are treatment and sex, so it's a two-way treatment structure. And blocks and reps are random effects. Here's what the pigs might look like in one of the pens. Put little tails on them there if you want. Put little ears on them. The snout. So anyway, I got big pigs. The blue ones are males. Big blue is a big one of the pigs that's in the big block. Remember, we blocked according to weight. And the pink ones are females. And we got 4 blocks of pigs occurring in this pen. This is the pen, say, that got treatment A. Then pens 1, 5, and 9 look just like this, except that treatment B is assigned to pen 5, treatment C is assigned to pen 9, and then each pig is measured over a 5-week period. And so in discussing the analysis, I'm going to try to make it a little simpler. I'm only going to consider the first 2 reps initially. We got reps 3 and 4 as well, but we'll only talk about the first 2. So we got 12 pigs of each sex. We got 3 pens, 2 pens of each type, of each infestation treatment, and we got 4 blocks of males and 4 blocks of females. And so that's sort of the treatment combinations that we observe, identifies each pig according to what rep it's in, what pen it's in, what sex it is, what treatment it gets, what block it's in. So when I see something like this and I have no idea what to do with it, I start thinking about contrast. What effects can I measure? And so if I look at the comparisons, say some comparisons between the treatments, then I have 2 orthogonal contrasts labeled here, B versus C, so infested and treated versus uninfested, or infested and not treated, and then A versus the average of B and C, so the control to the 2 pens that got infested. And so the pluses and minuses in there would represent the contrast that would measure those particular effects. And then I also have a contrast over here that measures the differences in the 2 reps. And then looking at the pluses and minuses in those columns, I can see that the pen effects do not cancel out. And so those contrasts are confounded with differences in pens. And so the treatment structure is over here in the B versus C and the A versus B, A versus the average of B and C. The blocking structure has to do with the 2 reps of these different treatments. And so the error for that comparison is going to be the interaction between the treatment effects and the rep effects. And so there's 2 orthogonal contrasts there, and the sums of squares corresponding to those 2 orthogonal contrasts would make up the error for treatments, for comparing the 2 treatments to one another. If you look at the, if you include in that as well, in addition to rep, include the sex and the rep by sex interaction, then you can see that those effects, the block effects don't cancel out. And so those particular contrasts also contain block-to-block information. And so if you write out the orthogonal contrasts in blocks that are orthogonal to this group of 3 here, then we got 8 blocks, so altogether there's 7 degrees of freedom to work with. 3 of them are defined by this part over here, so I got 4 more. And so the 4 that I've identified over here are orthogonal to each other, and they're orthogonal to the 3 that I put out over here. So those make up the interaction between those and sex would make up the error for comparing sex to one another. And then if you look at the sex by treatment interaction contrast, it ends up being free from both block and replicate effects, and so the error is all that's left. Well, it turns out that actually once you know all that, you can write out a SAS model that will analyze this data. You can write POC mix, class, rep, sex, treatment, block, the model Y equal treatment, sex, sex by treatment, and then include rep and rep by treatment, rep by sex, and block, all in the random statement. And it produces the ANOVA table that's shown here. And so I have the replicate effect, 3 degrees of freedom, treatment with 2. This is for the full 4 reps. And rep by treatment interaction is 6. And that makes up the error for treatment. And then we have sex, and rep by sex becomes the error for sex. And then block, well, some of the block effects are in the 3 degrees of freedom for rep, the 1 for sex, and the 3 for rep by sex. So altogether we have now 16 blocks, and so we used up 3, 4, 7 of those, and so there's 8 left over that haven't been identified in the previous part of this ANOVA table. So those all together make up the 15 degrees of freedom for comparing blocks, and we have sex by treatment interaction, and the error for that is what's left over. So it's got 22 degrees of freedom. So we've got lots of information for testing interaction. We don't have very much for testing sex, only 3 degrees of freedom for comparing sexes. We don't have too much for comparing treatments, only 6 degrees of freedom for that. So then the question is, I guess, is there a better way to do this experiment? I would hope so, but I don't know what it is. I'll let you worry about that. And the other thing is we now have to take care of the 5 weekly repeated measures part of the experiment. Good news is that's your assignment. Send me your answers by 10 o'clock tonight. Thank you very much for coming.